She did her undergrad at the University of Georgia at the Odom School of Ecology for dogs. From there, she went to Eastern Illinois University, where she got her master's and studied aquatic snake-prey interactions. And then returned back to UGA at the Warnell School of Forestry and Natural Resources to get her PhD, which is where I met her in 2018. And believe it or not, like all those years ago, was a bright-eyed, bushy-tailed freshman looking to get into salamander research, and Cindy had her dissertation project that I just latched onto, and we've been friends ever since. So with that, I'm going to turn it over to Cindy.
Thanks for having me. It's exciting to be here. I said several times today, I always forget how close Clemson really is to happen. It's really hard. We should do more things together, although I think we just did a bio blitz. And I was on a field trip with my class during it, though, so I don't know who won. Do you know?
It's undecided, really.
Oh.
Yeah.
Interesting. All right. But I'm happy to be here with you to talk about some research. It's a nice break from sort of hectic end of the semester teaching. I am in a faculty position in Warnell. I am a lecturer, but I also have an undergraduate research program and teach classes like herpetology, wildlife science, which is our kind of study design and wildlife techniques course, natural history of Georgia, and then some non-major classes like natural resource conservation. But I am a herpetologist at heart. And like Kevin said, I started out in the snake world and then transitioned into the salamander world, and now I straddle several horses. But I'm going to talk to you a little bit about some of the research that came out of my dissertation and some kind of ongoing projects that we've got brewing in the lab right now. And so I'm going to give you some basic overviews about salamander sort of ecology and biogeography at the beginning. But so essentially this talk is about hybrid zones. And so often people love to use this phrase that hybrid zones are the windows into the evolutionary process, or they're natural experiments to understand the evolutionary trajectories. And it's a overused phrase, but it's a pretty good one. And so often when distinct lineages overlap, they can produce sort of individuals with mixed ancestry. And it allows us, when this happens, to see some evolutionary mechanisms in action, and to kind of begin to tease apart how selective forces have shaped traits among lineages and make predictions about how they might react under future changing conditions. And we know now that hybrids are much more common than we once thought they were. And I think we probably still are far underestimating the number of species that do hybridize. This. Oh, we're going to talk about that. So yeah, this natural laboratories piece comes up a lot, but I think it's fair. So in the southeastern US, we are fortunate to have a lot of salamander diversity. We have maybe not the top rank in terms of just overall richness, but we've got a lot of species that co-occur and kind of pack in together into habitats. And much of that diversity is contained within this family called the Lepidontidae. And these are lungless salamanders that are terrestrial. So they bypass the traditional amphibian sort of life cycle of having an aquatic larval stage, and instead their offspring go through all of that metamorphic kind of process within the egg and then hatch out as miniature adults. And so they're freed from this relationship with water that many amphibians have to grapple with. So they're more terrestrial, they have no lungs, they breathe through their skin, and we have just tons of them in the southeast and in the eastern US. And in many cases, we see that they partition habitat along elevational gradients, and that they have these areas of sort of sympatry and allopatry where they sort of cross each other's paths. And in some cases, in places where those lineages come into contact along those gradients, they hybridize. And hybridization in the plethodonids is a very messy, uncertain area, because we have these interactions of lineages that are happening across very different timescales. So we have some species now that we think are species of hybrid origin. So they represent hybridization long ago. Then we have lineages that are currently hybridizing. Some of them are those that came from hybrid lineages themselves. And so teasing apart what the relationships are between all of these lineages is a real nightmare. We fortunately have some very, very talented, brilliant folks working on trying to untangle those things right now. And so we should be getting some interesting results on that soon to see kind of how wrong we've been about some of these relationships. But the situation here is complex. All right, and so one of the places where one of these hybrid zones occurs is the Coweta Basin, which is in the Nantahala National Forest in Macon County, North Carolina, western North Carolina, just right across the border in Georgia. And this is a large basin site that's run by the Forest Service. And it is a place where two species or two lineages of plethodon kind of co-occur and hybridize. And so in this forested watershed, there's a pretty substantial elevational gradient. And at the more montane high elevation site, the predominant lineage is plethodon shermani. And so this is a species that's characterized by having bright red legs and a black body. And the sites they live at tend to be a little bit cooler, a little bit wetter, and they're relatively abundant in those places. Then we also have a lowland lineage called Plethodon teahali. And this species is characterized by its white spots. I point these traits out because they will be important later. And they live in these drier, low elevation sites. Dry is relative, but compared to the very wet sites, they are quite dry. And then at intermediate sites, you have hybrids. And those hybrids possess a range of traits from both of those parent lineages in all kinds of combinations, any combination you can imagine. All white spots and also all red legs, or just a little bit of either, a little of one, a lot of the other. We get kind of every single potential option. All right, a lot of people have been interested in this system. So this Shermani, everywhere it occurs, it also occurs with teahali and also hybridizes with teahali. And so there are several isolates where these systems occur in this general area. And folks have been really curious and interested about it for a long time. So Harrison has several papers where he's sort of exploring this relationship between these hybrids, trying to characterize this hybrid zone and figure out how long it's been there, what caused it to occur, what's going to happen to it. And so over 16 years, he kind of looked at where he was finding individuals with different sort of phenotypic traits across the basin. And in this study he published in 1992, he found that pure Shermani, so what we would call individuals that have red legs, but possess none of the white spotting traits we see from the other lineage. So places where we see those, those places were shifting upward. And so at places where you once could find those pure teahali, they were declining, and that they were then present in sort of higher elevation areas. So they're kind of moving upward. And then in 2009, Susan Walls reanalyzed his data set because she was kind of skeptical, I think, about exactly what was happening there, if the Shermani were actually declining or if they were moving up, and what was the factor that was causing this to happen. So it was happening along an elevational gradient, but we don't think elevation is the biologically relevant factor here that's operating the system. And so she thought climate was likely to be important. And she found that there was this sort of upward shift in the system with rising temperature. And so at the time at Coweta, they had kind of very coarse ways of measuring temperature and precipitation and other environmental factors sort of across the basin. But using the limited sort of weather stations and data that she had, she was able to kind of establish that relationship. In 2016, there was a student who worked on his dissertation again in this system, and he was really interested in trying to see, is this a stable system? It's very old. We know it's quite old. There's some debates about how old it is, but thousands of years old, we think. And is it a stable system, or is it moving in a direction where hybrids are going to swamp out either of those parent lineages, or vice versa? And because there are these concerns, right, that the zone is shifting around, is it actually that they're sort of subsuming each other? And what he found is that the hybrid zone itself is much more narrow than you would expect based off of kind of a prediction of neutral diffusion through these lineages, and that they're not just freely merging and eventually going to consume either endpoints, that actually this is a pretty narrow zone of hybridization, and it seems to be held stable and in tension. And he also found that support that the mechanism holding it in this somewhat stable state is likely to be something environmental. And so the important thing here is that these lineages are not rapidly merging. They've been this way in this system for a long time. There are aspects of sort of where that system is situated along a gradient that seem to be shifting around, but that overall these endpoints seem to be being maintained. But that climate is clearly important. And so one of the things we want to know is sort of on a smaller time scale, kind of how is climate going to impact the way that traits move from one lineage into another, the physical location of this hybrid zone, sort of what the strategies are that promote the maintenance of this sort of swarm of individuals in the middle, and what keeps those endpoints from sort of diffusing in. All right. So the first thing we want to know is just kind of how can we untangle what the selective pressures are that are shaping the system? So which hybrid traits are they acting on? What's important? And so some potential traits we thought might be under selection here are morphology. So things like coloration, obviously pointed out there are some big differences there. They're quite striking. Body size is another thing. Physiology, so their things like their resistance to water loss, their thermal kind of gradient over which they can persist and be active, and then also their behavioral responses, so things like when they're surface active, when they're foraging, what their opportunities are to engage in things like courtship. All right, so this project has quite a few elements. We're going to spend a little bit more time on some of them than on others. But so the first part is really kind of the foundation of the whole project. So over time, we've had lots of people looking at the system, and what they've done is looked at the amount of red on an animal's legs and the amount of white spotting, and then put those into kind of bins and use that as a proxy to estimate what proportion of each parent lineage an animal is. So if it has intermediate presentation of a Shermanite trait and an intermediate presentation of a Teaholi trait, then people would infer that that means it's maybe a 50-50 hybrid. And if you have a lot of Shermanite and just a few white spots, then you're probably, mostly 80% Shermanite, 20% Teaholi. But we don't really know what that relationship is, but we've been using those phenotypes as a proxy for that ancestry. And so the first thing we wanted to do is just kind of see what actually is the relationship between these two things. Does how you look actually predict what you are and where you came from? Then we were also interested in looking at behavioral differences. So these salamanders, their ability to forage, to grow, to find opportunities to mate, are all dictated by their ability to be surface active. If they cannot be out and active on the surface at night, then they cannot engage in those activities which are critical for them. And so we were curious about how activity differed among different groups. So in animals that were appeared to be entirely Teahale or entirely Shermani, and then at various places in that hybrid zone. And so we used pit tag telemetry for that to be able to find them underground. They spend a lot of time underground, so we needed to be able to differentiate, are you just not surface active, or have you actually left this plot? Because they can be, just a foot underground, but in terms of your assumptions, it's an important thing to be able to differentiate. Then we also had kind of a wacky idea about measuring water loss after we really struggled to get permits to collect enough individuals to bring into the lab to do sort of more traditional methods of measuring water loss. So North Carolina kind of has been steadily cracking down on permits they issue in the Nantahala National Forest, especially for anything that involves you taking those salamanders off property and then either euthanizing them or trying to bring them back. They kind of don't like either of those options. So we pivoted to trying something that was less invasive but was unproven, which is to use a piece of equipment that is used for dermatology to measure your kind of water loss rates of your face. A friend of mine, Emily Taylor from Cal Poly, got this to measure lizard water loss rates. I think the Navy bought it for her, or maybe the Air Force, but one of her spendy DoD contracts got it, and I convinced her to bring it on a plane to North Carolina and use it on some salamanders. And so it was interesting to try out. I think it's not quite there yet in terms of being a perfect method, but it's definitely an interesting approach. And then another thing we were interested in is looking at body size at maturity in these animals. Like many things, they grow and grow and grow until they reach sexual maturity, and then their growth slows down considerably as they then allocate their resources towards reproduction and just for survival in general. And so we were curious about body size and how that might interact, how they the pressures they experience from the environment. I'll talk a little bit more about that at the end. All right, so here's the Coweta Basin. This is showing you kind of a precipitation and elevation gradient sort of all combined and laid in here, but you can see down here is sort of the lowest area of the basin that's the driest. And then as you go up this way, you're getting cooler and you're getting wetter. But we've got north facing slopes, south facing slopes. We've got areas that are coves, we have nice flat areas, very steep areas. So lots of topology, and then topography, and then we've got various kind of watersheds throughout this system. But we can kind of think of dividing this basin into two areas. We call this the Ball Creek sort of side, and this is the Shope Fork side. And there are roads that go up either, but they are kind of distinct. And they fall out sort of genetically distinct as well. And all of these red dots are sites where we've collected some portion of data that I'll present to you today. All right, so the first thing is, how are genomic and phenotypic traits distributed across the hybrid zone? So we sequenced tissues and we developed this genomic hybrid index, and using this program called GG Hybrid. And then we created these models of genomic declines to look at patterns of introgression in this system. And then looked at this relationship between hybrid index and phenotype. So how well does how you look predict what you are? And then how do those hybrid scores kind of vary with respect to aspects of climate? So we caught a ton of salamanders throughout the basin. We collected tissues from them. We got things, measured their phenotype scores, and then released them back. And so just quickly, the scoring system. So each animal gets scored on an independent system. So for the Shermani scores, we're looking at how much red is on the legs of those animals. And so they get a score of 0 if they've got nothing. And then the next barrier they have to meet is at least 1 dot of red on their legs. And then that goes all the way up to a 3, which is we call sort of full presentation of the trait, which is continuous red from shoulder to wrist. And so if you can sort of trace a line from shoulder to wrist, then you get a 3. And then for Taya Holly, a 0 is no white spotting at all. A 1 is just any amount of white spotting on the sides. But then 3 is fully presented trait. You've got white spotting sort of continuously along the sides and then also on the back of the animal. All right, so this hybrid individual, if we caught this while we were out sampling, it would receive 2 scores. So the Sherman score is that it's got continuous red from shoulder to elbow. So it only rates a 2 on the scale. It would need to go all the way to the wrist to be a 3. So it's got a Sherman 2 score. And then the white spotting, again, is present along the sides. It's more than just 1 white spot, but it doesn't continue on to the back. So it gets a 2 as well. So everybody gets these scores. So we prepare these 2 rad libraries, we sequence them, we demultiplex them, then we create these genomic hybrid indexes using ggHybrid. And then we identify these outlier loci with biased introgression. So we're interested in, is there asymmetrical introgression of some of these loci through this hybrid zone? Are there some traits that are moving sort of faster through the system than you would expect, sort of based on random diffusion alone? So we're looking at kind of allele frequency versus hybrid index at each locus. Okay, so genomic lines show you this change in allele frequencies at a particular locus across individuals. So you've got locus allele frequency on the y-axis and hybrid index on the x-axis. And then the Klein center is located where the hybrid index, where those 2 frequencies are equal. And then if you shift the center of that, then alleles are introgressing further into kind of the other side. So it can be biased in either direction. So we were looking for outlier loci that had these biased Klein centers that showed us they were doing something different than what we would kind of neutrally expect. And we had some outliers in both directions. So they existed kind of across the board. But the interesting thing we found was that so there were 365 Taya Holly biased outlier loci, and then 104 for Sherman. So disproportionate bias towards Taya Holly alleles kind of moving through the system. And another graduate student named Sam Weaver did something kind of similar with a different data set and found generally the same pattern. So in other places where we have these Sherman Taya Holly systems, we seem to see evidence that there are some Taya Holly traits that seem to be introgressing through this hybrid zone at a faster rate than what we would expect if there were no kind of selection pushing that process along. So it gives us a hint there's something going on here. Unfortunately, we don't have a reference genome for these lepidonids, and so we can't pick out any of those and say, oh, what trait is this linked with? What is this trait that's under selection that we see this pattern with? It would be amazing if we could. Some bird folks have been able to do that with reference genomes. I think some fish people too. I'm infinitely jealous. I hope at some point soon we will get to the point where we can do that because it would be really, really exciting. But for now, we just have to kind of speculate and wait. So then we're interested in what's the relationship between the genotype or the ancestry and the phenotype in these salamanders from the hybrid zone. So here we have a bit of a bizarre modeling approach. So this data is, it runs on the hybrid index score data. It goes from a score of 0, which is completely Taya Holly ancestry, to a 1, which is completely Sherman ancestry. And everybody's scored in between there. So we have this data that's bounded by 0 and 1. But we also have lots of individuals who are 0s and who are 1s, which creates a real nightmare for figuring out how you're actually going to model and use that data because you've got this concentration on the ends, and it's very hard to kind of find a distribution that lets you have that kind of a setup. So sometimes people will scooch their values in to kind of cheat and have them not be right at the bounds. But there are a lot of reasons why you probably shouldn't do that. And so we really struggle with figuring out how to model this data. The other thing is the phenotype scores are ordinal. And so you should know, everyone should know, I think this is one of my soapboxes, that you cannot treat ordinal data like it is continuous data without sort of acknowledging the potential pitfalls of that and the downsides of that. So overall, really a tough data set to work with. So what we landed on is using a Bayesian Zoib model, which is a 0-1 inflated beta regression model in a Bayesian framework using STAN. And so each score here is kind of modeled alone and then together to look at their impact on, or how well they predict hybrid index. So just looking and seeing how, if we only look at Tail scores, how well does that predict hybrid index? If we only look at Sherman scores, or if we put them together. And so I'll skip to the good part, which is that our best model is an additive model where you include both scores. That tends to do the best job predicting that hybrid index. And we use site as a random effect here because we have many individuals collected from single sites. And so we kind of want to account for that. And so I'm going to show you some predictions based off of those models. And I just want to say that, so we have site as a random effect here, and it tends to be important. But the predictions I'm going to show you kind of marginalize across all of the sites. So they give you a view of kind of an average site, which we can all wonder what that means, but of an average site here, what you would expect to see. So this plot requires a little bit of explaining. So over here on the y-axis is going to show you this hybrid score index. So a score close to 0, more Teahali. A score close to 1, more Shermani. Then along the x-axis, you're going to see the Shermani score level. So a 0, 1, 2, it's cut off a little bit, but then 3. And then we're going to plot distributions at each of those score levels for each of the Teahali scores. So we've got 16 combinations of things we need to look at here. And so this is it took me a really long time to figure out how it was possible to do this. And so each of those Teahali scores will show up as a different color. So I'll walk you through what this actually looks like. Okay, so here we have individuals where they have no Teahali traits at all. These have no white spotting for any of these. And it's showing you the combination of, for individuals with no Teahali spotting, what the distribution of predicted hybrid index scores are for each level of Shermani. So for an animal that has no Shermani traits and no Teahali traits, this is the distribution of hybrid index scores you would predict to see across an average site. And then if you have a Shermani 1, then this is your distribution, 2, 3. So you notice there's really not a whole lot of difference there going on. Those sort of are relatively consistent in their shape and location. You get kind of a little bit of shifting up towards being in more Shermani territory as you have increased presentation of Shermani traits, but nothing really dramatic that you see there. And so then, all right, so now we're looking at if you've got very low presentation of Teahali traits, just some white spotting. And so then again, we get just a little bit of Teahali signal, same shape in general, and not really a big kind of impact of that Teahali score necessarily there. Then we add in the 2s. So now we're getting much stronger presentation of Teahali traits, and we get a bigger shift here. So the ridges get a little wider, especially when your Shermani score is high. You sort of tend to lose some confidence in exactly what hybrid index score you would expect an individual looking like that to have. And then our kind of biggest shift here is, so when we have the strongest presentation of Teahali traits, then we kind of get more confidence that when you are pure Teahali in your phenotype, so you have no Shermanite traits and you only have the strongest presentation of Teahali traits, then we become the most confident we are anywhere that your hybrid index, right, is somewhere in here, probably around there. But you can see these ranges for these other kind of combinations are really not very predictive. They span, can be from 25% to 75%, which is, what does that mean? Tells you almost nothing, right? And so, as the Shermanite score increases, your uncertainty kind of grows, and there are a lot of places in the basin where we get a wide range of scores for animals. So at a given site, you can find animals that range from a 3-3 to a 0-0. It's not usually common that you have both of those extremes, but we do see them. And then we get all kinds of mixes of these intermediate scores. So when you zoom out and you look at the basin as a whole, the relationship between your two scores does generally tell you something about what your ancestry is going to be. What we found is that when you zoom into the plot level and you're looking to say, okay, all of these individuals in this plot have different phenotypes, they look different. Does that mean they actually have different ancestry? That was one of the things we wanted to know. And the answer appears to be no, that differences in appearance within a site do not correlate well with genetic differences. They're not very predictive. But again, if you kind of zoom way out and you go, oh yes, you have red legs and no white spots, you're probably mostly Shermanite. And we're even more confident of that if we know you're from a high elevation site, which you must be because that's the only place we find you. But this was interesting because this question about how they look in a plot, is that actually predictive of anything to do with ancestry? People have been grappling over for a long time. Some people I know are still unhappy about this finding because they truly believe in their heart of hearts that must be real differences, but we found really no compelling evidence of that so far. Oh yeah, just one little note here is that the within site variation in hybrid index scores is very, very tiny on average across this hybrid zone. This is a system that has been in this tension zone for a long time. So we don't have any F1 hybrids. There are no places where pure Shermanite and pure Teahali interact. They are all separated by this hybrid zone. So no new hybridization, and they've existed in these systems for a very, very long time. So it's not surprising to me that the variation in hybrid index scores within a site is very small on average. All right, so across the hybrid zone, phenotype tracks ancestry generally well, but at the landscape scale, at the small scale, within sites, it does not. Within site variation in hybrid indices is very low. If I know where you're from and I've looked at calculated hybrid index for somebody else where you're from, then I can reasonably predict what you are. But we do see a lot of variation in phenotype. And all of this is consistent with what we kind of expect to be going on and what Lowe found in his dissertation, which is that this is a system that is being maintained in this state, and likely that it's this exogenous environmental selection that is creating this pattern. So now our question is, what are the environmental gradients that are sorting ancestry across sites? And Susan Walls and Harrison, they found this evidence that there was some movement in the hybrid zone. And so how are those climatic or environmental factors potentially impacting this hybrid zone on the short term in terms of moving animals up and down, but also long term in terms of their kind of persistence in this area and the relationships between them. Okay, so we were really curious about how hydroclimate in particular impacts the distribution of traits. So we used, again, a Bayesian 0-1 inflated beta model using Zoid to model this relationship between factors we know are likely to be important. So precipitation is the one I'll show you today. We've also looked at vapor pressure deficit. You can go back and redo what Susan did and use temperature. Precipitation, temperature are highly correlated in this system, so they both do kind of an okay job explaining it. But we think that this piece about precipitation, about water, is really the most biologically relevant angle. Temperature really doesn't matter if your water balance is not conducive to existing. We also looked at some additional factors like slope and aspect, so how much sunlight a spot is getting, and then how quickly water drains through the soil, so how dry the soils are, as another potential factor that might be important. And again, we accounted for the fact that we had multiple individuals from these sites by using site as a random effect. All right, so now we've got, again, hybrid index on the Y, and then mean annual precipitation on the X. So very dry on one side, very wet on the other. And so when we sort of predict this relationship of what is your hybrid index likely to be based on mean annual precipitation at the site that you live in, we get this kind of pretty nice little pattern. And so these dots down here are from what we deemed our parental sites, so places where we thought animals were as close to being pure Teaholi as we could find adjacent to our site. And so they show up with their very low hybrid index scores. And then up here are pure Shermanite sites, again, very close to 1. And then all of these are from the hybrid zone. And one thing you might notice is that mean annual precipitation seems to do a pretty good job, except there's something weird happening here, right? That these two sites have the same mean annual precipitation, but they have vastly different hybrid index scores at those two sites. Which might make you think precipitation maybe doesn't do that great of a job of predicting this, because it doesn't really explain that pattern. We managed to kind of scooch in between there. But so this site is very interesting, and it's a really important one to have in here because it is a high elevation site that is drier than you would expect based on how high it is. So it breaks this relationship between precipitation and elevation that we have a really hard time getting away from in this system. And interestingly, at this site that should have individuals that look like this, just based on the elevation, when you look at precipitation, it makes much more sense. They are very, very Teaholi in their appearance and also in their ancestry. We don't have currently much data from other places in the basin that break the rules. And so that's something we really need to, in order to build, I think, a model that captures this relationship a bit better. But it does kind of allow us to see this piece about that precipitation is likely the best variable to use to capture those differences across those sites that we think actually are driving patterns in ancestry. Okay, so then our next question is, what's the relationship between phenotype and climate? We wanted to chase it from this side as well. We have a lot of individuals that we have phenotype data on, but not genetic data. So these results are going to be ugly. So just bear with me for a second, because for this ordinal data, we've got to estimate kind of probabilities for each of these individual scores. But what you see is that the likelihood of having a score, a given score based on the precipitation at that site is kind of what's modeled here. And so as you get into wetter areas, the likelihood that you have an animal with high Shermanite trait presentation goes up. And as you go to drier areas, the likelihood that you get no presentation kind of goes up and then goes down. and intermediate presentation. And then no presentation steadily increases. So none of it is really groundbreaking, flabbergasting. It's the same kind of relationship we would expect, but we can see it very clearly tracks with precipitation. So how you look, again, at a broad scale does have some indication of where you will be found, and a little bit of who you are. So we get kind of the reverse relationship for tail. So we get this relationship kind of on both sides. So just to sum up a few things. So we've got biased introgression of alleles from both lineages, but more of them from Teaholi. So we think those Teaholi alleles are under active selection. We don't know what they are. We hope we will know someday. Phenotype score is generally indicative of ancestry at a broad scale, but not so much within a site. And that mean annual precipitation is a very strong prediction of hybrid index. The relationship between precipitation and phenotype is slightly more complex, but again, still pretty evident. All right, so briefly, I just want to talk a little bit about this behavior piece that we've done, which is looking at surface activity. So we put pit tags in these salamanders and we tracked them around their plots for kind of 2 seasons. And we were interested in how weather impacted surface activity and the relationship between ancestry and surface activity, and trying to kind of estimate survival and detection. And so we used a multi-state Marbury capture model, a CJS model kind of adapted in JAGS, and we looked at hybrid index, 3-day rainfall, temperature, and body size as potential important predictors. So we had 126 individuals. We tracked them for almost 90 days and got lots of detections. Nobody died that we knew of, but we did have 107 individuals that we only ever were able to re-find one time. So lots of wily salamanders that evaded us. All right, so here what we have is for individuals with different hybrid scores, what was the likelihood that they were surface active across a 3-day rainfall gradient? So we've grouped them into, these are the Teaholi guys, these are the hybrid guys, and these are the Shermanite guys, and they're from various sites. And so this is looking across that 3-day rainfall gradient, which we found to be very important. And so the vertical lines on there are the solid line is the mean 3-day rainfall for that site. The max is the dashed line. And so we can see that surface area activity or surface activity increases with rainfall here. But we see really no meaningful differences between sites. And we do see differences in the rainfall conditions that are experienced at each site. So basically what we're saying here is that under a given set of conditions, we expect all of these salamanders to have the same levels of surface activity. But what's different is the conditions they actually experience given where they are. So Shermanite, we think of as being more surface active, as that just being a trait that they have. But really, their propensity to be active is just the same as anyone else's. It's just that the opportunity is greater. And then the reverse is true for Teaholi. So in this case, a salamander is a salamander is a salamander. And just depending on where you live, you may have more chances to actually be out and about. So yeah, we saw really no differences in the likelihood of surface activity across sites when we controlled for which site we were at. The probability of surface activity increases with increasing rainfall, just across the board. And when we incorporate ancestry, does the probability of being surface active differ? So this is a place, again, where we're looking at an average site, which again is just a theoretical concept. You can see that probability of surface activity versus 3-day rainfall. There's no defendable differentiating between those estimates. I do know some people have tried to do some reciprocal transplant experiments with plepidonids, and it does seem like this might be an opportunity to do that, although it makes me quite nervous. But I might contaminate my own hybrid zone on accident, but it would be interesting to see. So again, rainfall, best predictor of surface activity. We did find an additional positive effect of body size. So this is all about surface area to volume ratio. If you are a bigger salamander, then you will lose water at a lower rate for your body size than if you were small. If you're small, you have a lot of surface area and you're just shedding water all the time. And so we know that. And so it makes sense that this relationship is clear there. We would be shocked if it weren't. But again, no differences in surface activity among sites if they were experiencing similar weather conditions, same thing for lineages. And so all of this is just about differences in rainfall at these different locations where we find individuals of these different groups. All right, so the water loss rate from the skin piece, the cutaneous water loss, I'll give you just the cliff notes of this. But we had 140 salamanders from 6 sites. We acclimated 61 of them for 7 days beforehand, just to kind of see if there would be any difference if you pulled them directly from the field or if you let them all acclimate to the same conditions, regardless of where they were from. There's been some really cool work by Eric Riddell about plasticity and water loss rates in salamanders. And so we were a bit curious about how the conditions you're experiencing influence your water loss rates. And so we standardize those when we see a different pattern. So we used a linear mixed effects model, and we included a bunch of covariates about temperature, whether they were acclimated, how big they were, and then hybrid index at a site and median annual precipitation. And so the results are pretty straightforward. Water loss rates increase when the temperature increases, which makes sense. It gets hot, you lose more water. Rates decrease with increasing body size just across the board. And then when we look at individuals of different hybrid indexes, we find that there's really no detectable difference in their water loss rates. So their capability to hold on to water appears just to be the same across the board. And then the last thing is body size. And this is probably the most interesting thing, but it's something we're kind of working on right now, flushing out. But this body size piece we know is important for water loss. We see differences in body size kind of overall, with Teaholi tending to be a bit bigger, more robust than Shermani. And so you're kind of curious about that pattern. So we have lots of capture data from lots of animals over a long period of time where we have sexed them, and some animals where we have known aged animals that we've sort of traced through their adolescence into the time when they become sexually mature. So we're kind of interested in seeing, do they delay maturity at these drier sites to give themselves a bit more time to allocate resources to growth instead of shifting over to reproduction. Once you make that shift, that's sort of the body size you get. So if you can delay, you can be bigger. So we had just over 1,600 confirmed reproductive adults. We have 51 combinations of site and sex here. We just ran this in a linear model. We looked at sex, median precipitation, and then slopes of the kind of steepness of the site that they're from. And so in general, the minimum observed size at maturity, so the smallest size where we found individuals that had reproductive structures that we could definitively identify, was negatively correlated with mean annual precipitation. There's a little bit of difference between sexes. And then it was positively correlated with slope. And so animals mature larger in drier areas, and steeper slopes kind of drain water faster. So that's where we get that kind of positive correlation there. And so we predicted that there would be a 20 millimeter increase across a short rainfall gradient. And one of the problems we have is that we're kind of limited in how we can make inferences about this, because we don't have a lot of growth data for animals in drier areas. They are harder to find. There seem to be less of them. They seem to be kind of not as concentrated in the environment, and the conditions in which you ideally will find these salamanders just occur less often at those sites. So we have kind of a hole in our data there in terms of getting some measurements from animals from dry sites. And so that's kind of a problem. But based on those mid-zone curves where we have lots of data, we think that 20 millimeter difference in size that we see across that gradient potentially takes 3 to 5 years for those salamanders to accumulate. So it's a pretty significant delay in becoming a reproductive adult. So you can imagine there are lots of demographic consequences of following this kind of life history strategy. And so that delay in reproduction may be really important. And when you think about Shermani at the top, right, they can be smaller. They don't have to worry as much about water loss because they're in a rainy place. They can start reproducing much earlier. And so one of the things we've been kind of considering is, could Taya holly have an advantage in that it can persist in these dry environments in a way that Shermani really cannot, because it is following this pathway of sort of not growing as far, but then reproducing very early. And then can Taya holly really kind of not hack it in those wet environments where they're getting demographically outcompeted by pure Shermani? And so is this maybe part of a mechanism that's holding those ends in the place that they're at and preventing too much introgression into those endpoints? Is that those kind of very different life history strategies are really only effective at those extremes. And so it's something we're kind of digging into. We've just looked at the Coweta data set here, but got a graduate student right now who is looking at a much larger data set of plethodon throughout the region and trying to see, can we pick up differences between sites, between species, and what kind of overall patterns can we pick up there. But it's very interesting. So I've already told you about this. Skip ahead. Okay, so we really wanted to look at these genomic and morphological declines and then evaluate differences in behavior, physiology, life history, all of that stuff. And so we know that water in the environment has an important impact on surface activity of those salamanders. That's a big thing that's limiting whether or not they can go about the business of being a salamander. And body size has a big impact in that as well. And so we know that the relationship between that genomic variation is tightly linked with that hydroclimate. And again, size at maturity is also linked with hydroclimate. Certainly there are other aspects of those environments that are important, but these are the ones that have kind of provided us the best explanatory power with our current data set. We didn't find any differences in surface activity to kind of compensate for variation in climate. So we don't see, evidence that Taya holly has begun to kind of develop increased resistance to water loss to account for the fact that they have less opportunity to be surface active. It's just that they grow bigger and weather that dryness a bit better and spend more time underground. And that we don't see any kind of physiological differences in their water loss rates. So they are a salamander at the end. And so in the absence of any kind of compensatory behaviors or physiological compensation, we think variation in body size might be the sort of most rapid response that they may have to dealing with shifts in these pressures imposed by changes in the environment and changes in climate. And so by delaying maturation, it gives them this toolkit for changing that relationship. And the really interesting thing is, right, we see this biased introgression of Taya holly alleles up through that system. What might those be? Might we see at some point that hybrids start to become a bit bigger as those intermediate sites become more xeric, they become drier, the rainfall patterns shift. Over time, will we see the things that make Taya holly successful move as the conditions that Taya holly experience also move up in those elevation gradients? And so it's very similar to kind of what has happened to these salamanders in the past, though on a completely different time scale. The Southern Appalachians were protected somewhat from glaciation. They were this sort of safe haven, but they also had a lot of climate variation. So salamanders were constantly getting sort of pushed to follow the conditions that they preferred, often up elevational gradients in times when it's hot, and then back down when it's cool. And so they get these periods of being isolated and then coming back together. And so we get all this hybridization. But so we know that they're doing this on a really, really long time scale. And so now potentially are we seeing them do this chasing up piece? But what's going to happen once they all kind of start to get compressed at the top? Some people think Shermani we will lose, and then we will just have hybrids in Teahali. There is always the possibility that we wind up with only hybrids. But it's pretty interesting. But yeah, climate is clearly important. Introgression is definitely increases the capacity for there to be variation across this gradient, which may give them a better chance at weathering the storm than species that are just left with their singular toolkit of their ancestors. And so one of the things that I always like to highlight that's important is that in general, hybrids have no legal protections. They are in many cases considered threats to pure lineages. And so they sometimes, in addition to not being protected, are targeted for removal, sometimes deservingly, sometimes not. But so right now, the salamanders that I study have really no protection at all. And they're fairly common right now, but that may not always be the case. And we have to kind of think about this question about what do we preserve, what do we value, how do we decide what we want to keep around, and what extent are we willing to go to do that. And my perspective on this has changed in that now I think about the evolutionary history of each of these lineages, and that I want that adaptive capacity they have to be the thing that's carried forward, right? Like I want salamanders to be in this system forever. I want them to persist. And if the best way for that to happen is for these two lineages to kind of share their best traits and come up with new combinations that allow them to do that, and it means we don't have any pure Shermani anymore, it's kind of sad, but also those traits live on. Shermani is still there. Just in the way that whatever hybridized to create Shermani in the beginning and hybridized to create Teahali in the beginning lives on in them now. And that we may need to kind of rethink how we make these decisions, how we define entities that we need to conserve, and all of these things. And so just kind of something to think about, but not a problem I see a resolution too soon, because it involves kind of government paperwork and decision making. And but yes, so I think any one message to you would be really keep an eye out for what's going on with hybrids in your study systems or in study systems you think are cool, because there may be really interesting opportunities to sort of look into these questions about what happens when you can break the rules and then you experience unprecedented times, as we all seem to be time now. But I'm happy to kind of take any questions or talk to you about my study system, but that is the kind of rapid run through of all of it. I couldn't choose a piece to drop, so but yeah, it's a fun system. There's definitely a lot still to do, and I look forward to kind of seeing what the next pieces are.
Time for one or two questions.
Yes.
So you talk about, as climate shifts, maybe we see Teahali traits being pushed upward, and it's a concern of comparison as well. But I also know that, like, over the past, couple centuries of fire suppression, we've seen mesofications throughout the southeast. Do you have any opinion on how that might be playing into the system?
Yeah, so there's yes, it's interesting. And so we have some experimental fire sites at Coweta where we've been introducing fire into places to see when you push them to be more xeric. What does that do to kind of the composition of individuals there or their behavior? And it does seem to have an impact on their surface activity, at least initially. We do see sort of burrow use change over time. And So I do think introduction of fire that disturbs that microclimate would have a pretty potentially significant impact on the salamanders, especially if you're introducing fire kind of on set intervals. This is something I think about with box turtles too. Makes me think of Kyle, which is that one thing I've been thinking about a lot is that in South Georgia, we have box turtles everywhere in all of these places, but a lot of them should be longleaf forests, and now they're kind of mesic forests. And then as we introduce fire and turn those into these longleaf savannas, box turtles seem to be not doing well in those places. But they shouldn't have been there to begin with, right? We suppressed fire and then allowed them to expand in there. And so it's a thing I think about at Coweta, which is that if we reintroduce fire at a natural kind of rate interval, and it is bad for the salamanders, is it really that we've just allowed them to kind of proliferate in the absence of that? And should we maximize on that benefit, or should we focus on restoring these systems to their what we think is their natural state? It's a tough one because there's kind of a lot of tension there. So, but so far we haven't found any signs that it's like very detrimental to them if we have long fire intervals. So we need a bit more time to figure it out. But yeah, that's definitely an unknown.
Yeah, I'm just generally interested in any evidence for diet preferences and how those change between hybrids.
Yes. So we don't have any strong evidence that there's any dietary partitioning among them. The size difference isn't really substantial enough to constitute sort of like a meaningful prey switching between the two groups. There are lots of little bugs at Coweta. And so, yeah, so we haven't seen any impact of diet, but certainly I think we don't have, at least in our lab, maybe the best resolution on how those invertebrate communities shift across the